let us begin sort of an overview of what happens during protein synthesis. So we have DNA. The DNA will specify the eventual structure of protein molecules. This polypeptide will eventually fold into a protein through secondary, tertiary, and possibly quaternary folding. So the DNA specifies that eventual protein. The proteins then will take on a particular shape and act as structural or functional molecules. Basically, they carry out the physiology that we discussed throughout the rest of the course. Anatomy and physiology, proteins are in charge of that. Now, proteins are comprised of these polypeptide chains, and the polypeptide chains are made up of amino acids. Each gene in DNA is a segment of DNA that carries instructions for one polypeptide chain. There are four nucleotide bases, which are, remember, the nucleotides are going to be comprised of three things: a ribose sugar, or I'm sorry, a pentose sugar, a deoxyribose pentose sugar, and a phosphate, and then that nitrogenous base. So most nucleotides are simply named for the base that they carry, that they have. The four nucleotide bases, A, G, T, and C, will be divided into a sequence of three nucleotide bases, and this sequence would be called a triplet. A triplet will specify a particular amino acid in the sequence of amino acids that eventually makes up the protein. So there are two main steps of protein synthesis: transcription and translation. So we're kind of showing in this picture an illustration of the central dogma of biology. DNA transcribed into RNA, pre-mRNA and then mRNA. And then the mRNA is translated into that polypeptide chain of amino acids that eventually becomes a protein. So the two main steps: transcription and then translation, with lots of little details in there as well. Transcription is the process of transferring information from a gene's base sequence to a complementary mRNA molecule. Translation is the process of converting that language inside of the nucleotides, the nucleic acids, the DNA, eventually into the language of the proteins. Now, as we can see in this picture here, to make an mRNA complement, the transcription factor mediates binding of an RNA polymerase, an enzyme that directs the synthesis of mRNA. Here we have the RNA polymerase. It sort of unzips the DNA. Here we can see the mRNA transcript kind of running along that and popping out. There's kind of a close-up of how this all works. Here's our RNA polymerase. We're creating our mRNA transcript by zipping along this particular gene on the DNA. And eventually, you will have the termination of the process, which will allow you to have the termination, will allow you to have a complete mRNA transcript. And this can happen multiple times. It can happen over and over to the same gene. It's actually really important for you to kind of, I know we're going quickly through this, and there's lots of details that we're not going over because they're not super essential here. But the idea of a transcription factor means that transcription will happen as many times as the transcription factor is present. As many times as that RNA polymerase runs over that gene and creates an mRNA transcript, that can happen over and over and over again. It just depends on how long the transcription factor allows that to happen. So one of the things, as we're kind of going through this, and especially if you're not familiar with the process, this is going to seem very speedy, like it's all happening really quickly. But the reality is that this is really how the body works. This is the essential basis of the way that cells actually work. It's through transcription that we start to have particular cellular behavior. And you can transcribe a gene many times, or you can down-regulate the gene to where you're not transcribing it as many times, or you can upregulate it to where you're transcribing that gene many times. So the amount of protein that you produce depends on how many times that gene is transcribed during that, period of time. I don't know, during growth, you might want to have a particular gene that is transcribed many, many times. And then as you get older, you might want to downregulate that gene and not transcribe it as many times. So, this is the basis of a process called epigenetics, where you can have very fast over the course of a lifetime or the course of just a few generations, you can have massive changes in a person's behavior, in their phenotype, the way that they appear, their characteristics, the way that their cells work, because it's not dependent on a gene undergoing mutations. It's dependent on a gene being transcribed many times or fewer times, for example. All right. So here what we're looking at is it's called a primary transcript. You'll see that here. You'll see a pre-mRNA transcript, and then you'll see the eventual actual mRNA transcript, and that is because you'll see that the mRNA transcript is actually shorter, because what happens with the pre-mRNA is that there are introns present. And what you want to do is you want to splice out those introns so that you end up with your actual transcript that you want. This will faithfully be rendered into a protein eventually. So the pre-mRNA is going to have a lot of DNA inside of there called introns, or in between the useful parts, introns, that will be spliced out and will be taken out, and you'll eventually have the actual mRNA transcript that produces the protein that you want. This pre-mRNA could, for example, produce several different proteins. So one gene could produce different proteins depending on how that original pre-mRNA transcript is processed into an mRNA transcript, and then eventually into that amino acid sequence, which becomes a protein. So the same gene could actually transcribe and translate into several different genes, several different proteins.